Our next speaker is Yujia Wang. Can you, Yujia Wang, I think, by the way, you have the same name with a great pianist. And I think you are from, Yujia is from the group of Professor Wei Zuo's group from Tongji University, and the talk she will give on behalf of Professor Wei Zuo is single cell analysis about the lung cells. I will not repeat the title. Can you share your screen?
Yes, of course. Can you see it now?
Yes, please make it full screen.
Okay. How about now?
Yeah, perfect.
Okay.
So I will remind you to stop at 10 minutes.
Okay, thank you, Chair. So good afternoon. So my name is Yujia Wang, and I'm from Professor Wei Zuo's lab in Tongji University. And Professor Zuo, he cannot attend this webinar due to some unexpected events. So on behalf of him, I'm going to introduce some of our efforts on the COVID-19 today. So the pandemic COVID-19 has caused around 40 million infections and more than 1 million deaths. So far, no specific antiviral treatment or vaccine has been approved by the health administration of any country. So today I will introduce some of our efforts in developing how the virus affects the lung cells and how the lung regeneration initiates after the viral infection. So now we all know that the cell entry and the subsequent receptor usage for the coronavirus, they have been studied intensively. And we all know that the coronavirus interact with the host cell receptor, ACE2, while its receptor binding domain in its spike protein. So understanding of the tissue expression and the distribution of the ACE2 is very important to study the virus infection. So we all know that the lung it is a complex organ with multiple types of cells. So it comes to us to use single cell sequencing to study the ACE2 expression. We responded very quickly and we launched a study on the distribution pattern of ACE2 in the normal human lungs in this January. It's shortly after outbreak of COVID-19. So this work is based on the public single cell database, and the results show that ACE2, they are intensively expressed in a small population of the type 2 alveolar epithelial cells. So these type 2 cells, they not only express viral receptor ACE2, but they also highly express many other genes. Those genes are related to the viral entry, the viral life cycle, replication, and also the transmission. So these cells, they may serve as an entry target to assess the virus transmission. And that was just the first step of our exploration. Which is more interesting to us is that since alveolar regeneration after acute injury has been observed in many animals, including human, and some other works has demonstrated that alveolar regeneration in COVID-19 lungs was initiated at a very early stage. So here is a report that they enrolled 2 patients, both showed severe diffuse alveolar damage. And the histopathology showed that around 1 month after the onset of initial symptoms, and some of the type 2 epithelial cells, they are able to replicate and also differentiate in other cell types, and therefore help the lung regeneration after the virus-induced injury. So as I just said, that there are multiple types of cells in the adult lung, and many studies has demonstrated that after the lung injury, there are multiple stem cells, or sometimes we call progenitor cell population. They are involved in the regenerative process. So our next step is that we analyze the bronchoalveolar lavage fluid of COVID-19 patients by single cell sequencing. So this work was also based on the public database from 3 patients with moderate COVID-19 infection and 6 patients with severe or critical infection and 4 healthy controls. So first, we performed the unsupervised clustering on the epithelial cells in the lavage fluid. We got 8 epithelial clusters, including the gallbladder cell, the ciliated cells, and also the mature alveolar cells. Also, we found a cluster of the lung progenitor cells. We found them by the expression of previously reported markers such as TM4, SF1, and P63, KRT5, and SOX9. Then we compared the healthy control group with the infected groups. We found that the higher proportion of alveolar cell is here. We found that the higher proportion of alveolar cell cluster and also the lung progenitor cluster, they exist in the severe infection group. So when we take a close look to the lung progenitor populations, we found that there's a subpopulation of type 2 cell. It is included in this progenitor cluster. So these cells, they are reported by previously reports. They are reported as a Wnt responsive population, and they are also marked by the TM4SF1 expression in the human lung. So we found that in the patients of severe infection, the number of this population also increased. So this means there may be a rapid activation of these cells. And we also observed co-expression of TM4SF1 and mature alveolar cell marker, the AGER, in the severe infection patients. And we think this means that the TM4SF1 progenitors, they had the potential to differentiate into mature alveolar cells in the COVID-19 patients. And also, according to the previously studies and also according to our own observations, the P63 KRT5 positive cells located in the basal layer of the distal airway, they are also reported to have lung stem cell features. So we found that in the patients of severe infection, the number of the KRT5 progenitors also increased. And also gene ontology showed that KRT5 cells, they were responsive to the low oxygen condition, and they highly express many other tissue integrity genes. And these genes were related to the epithelial barrier formation and also some kind of immune regulation. So we wanted to know that if the KRT5 progenitors, if their function is related to the barrier formation in the injured lung. So we test the idea on animal models first. Since the coronavirus, they cannot infect mice, so we used bleomycin to induce lung injury instead. So we isolate the mouse KRT5 progenitor cells, and we label the cells with GFP lentivirus, and we transplanted the culture cell into the injured lung. So the lungs, they were evaluated at day 30 and day 90 afterwards. So the results show that there is a substantial persistent engraftment of the GFP positive cells. And also immunostaining show that some of the transplanted cells, they can differentiate into barrier-like cells. Here we call them alveolar barrier cells. As we can see here, they formed a large area of sealed cavities, and it looks like they can prevent immune cell infiltration into the surrounding area. So next, we harvest and the transplanted lungs, and we sorted the GFP positive single cells for analysis. So clustering analysis identified 5 clusters, including the most immature P63 KRT5 double positive progenitors to the most differentiated cells. So there are 2 clusters that have lost the KRT5 expression, and they highly expressed early alveolar marker HOPX, and also some other markers related to tight junction. So this means this cluster might be the alveolar barrier cells. And also pseudotime analysis indicate that the original P63 KRT5 double positive progenitors, they can differentiate into P63 negative progenitors and in mature barrier cells, which eventually give rise to the mature barrier cells. So consistent with this, we notice that compared to the 30-day transplanted cells, the 90-day transplanted cells, they have relatively more mature barrier cells in their population. Since we got the evidence of the formation of the alveolar barrier cells on the mice models, so we mapped the data of the transplanted progenitors back to the COVID-19 data to see whether mature alveolar barriers has been formed in human patients. So the figure here shows that about 10% of the epithelial cells in lavage fluid of the severe patients could be annotated as mature barrier cells. So this is a whole picture of our findings. So after the infection and damage, the KRT5 positive progenitors, they can differentiate into barrier cells. And this helped to restore the new epithelial barriers to control the immune cell infiltration. And at the same time, the TM4SF1 progenitors, they could also regenerate new functional alveolar cells. So these two types of the progenitor cells, they could work together and restore the alveolar structure quickly after the lung injury. So future studies may be conducted within a larger patient cohort. Maybe it will give us a better understanding of the repair process of COVID-19 lungs.
It's already 10 minutes.
Oh, okay, I'll go through very quickly. And also it is feasible to grow the KRT5 progenitors in the culture dish. So it can be used as a potential progenitor cell-based therapeutic product. Now we have already tested on some chronic lung diseases, such as the obstructive pulmonary disease. And we have seen some very promising outcomes. And we plan to use it to cure acute lung diseases, such as the acute respiratory distress syndrome. So maybe someday in the future, the progenitor cell-based drug, maybe it can be used to help us fight with COVID-19. So here, this is a very brief summary of our work. We report the RNA expression profile of the ACE2 in the human lung. And in the severe or critical COVID-19 patients, there is a remarkable expansion of TM4SF1 and also KRT5 lung progenitor cells. We think that maybe the two types of the lung progenitor cells, they can work together to prevent and restore the alveolar loss after the virus infection. So this is all about my talk. Thank you. Thank you, Chair.
Okay, thanks. We have only 1 minute before we start the next. So any quick question from the audience? If no question, thank you, Yujia and